I think we'll move on to our next speaker, Dr. Eric Gendon, who is the Isidore Freisner Professor and Chair of Otolaryngology, Head and Neck Surgery. He is the Senior Associate Dean for Clinical Affairs and a Professor of Neurosurgery and Immunology at the Icahn School of Medicine at Mount Sinai. He's the Chair of Otolaryngology, Executive Vice President of the Ambulatory Surgery, Director of the Head and Neck Institute at the Mount Sinai Health Systems. I could probably carry on, but then we wouldn't be able to hear him talk. So Dr. Genden has been a pioneer in the otolaryngology field in many aspects of reconstructive surgery, from starts with pec flaps, free flaps, all the way up to transplants. And it's going to be a pleasure to hear him talk about our management of airway defects.
So yeah, thank you all for an opportunity. We'll switch gears a little bit and think about tissue engineering and regenerative medicine a little different. And it can be a very extensive talk because it's quite entertaining, the history of this. But for all of us, we're aware and this goes back over 100 years with the diphtheria epidemic in the early 1920s, 1910s that left innumerable children actually with defects in airway disease, but iatrogenic, burns, congenital malignancy, and now COVID, tremendous number of airway tracheal injuries. And the issue here is if they're in the cervical region, they're problematic. You can't speak, but if they're combination or in the thoracic region, they're often not compatible with life. And what's been interesting is, for over 100 years, this fascination with how do you reconstruct the trachea has been one that many have chased. And part of the reason is because it just looks like a tube. It shouldn't be that difficult. And in fact, in '66, in the early 60s, even before this, Hermes Grillo, who was up at Mass General for many years, did seminal work looking at the blood supply under the pretense that maybe we could transplant the trachea the way we do so many other organs. And his conclusions and many others in the 60s was that transplantation is not possible. There's no single blood supply. You can't hook it back up. And this dictum really defined medical research in this area and scientific research in this area for well over half a century. And of the 2,736 articles that are written about tracheal reconstruction, they all start out with, because transplantation is not possible, and then they move on with their engineered approaches. And if you look historically, even back in the 40s and 50s, they started using soft tissue, but this would collapse because it wasn't rigid. Under the minus 160 centimeters of water, this would just collapse. In the 70s and 80s, the introduction of plastics and alloplastics were used. Simple tubes. Problem is they didn't biologically integrate, and they eroded into the surrounding vascular structures, leading to catastrophic hemorrhage. And in the 80s and 90s, many of us tried using actually biological fixed trachea, just like you're using scaffolds with your bone. This was fixed trachea. The problem was there was no ciliated epithelium for these long segments, and as a result, these would get clogged. The patient would suffocate and inevitably die, and that was actually a very common problem with this type of reconstruction. And so part of the issue was people just looked at the trachea. It was a little bit the Rodney Dangerfield of organs. It got no respect. It was a tube. But when you look more closely at the trachea, it's actually an organ. And that organ has some very important components, just like a lot of what we deal with. The cartilage, but more importantly, the ciliated epithelium that moves all the particulate matter out of your lungs is critically important. And in order to have that survive, you need vasculature down below. So you cannot plant, as I say, grass on cement. You need a fertile soil. And that soil is the vasculature that sits in the trachea. And you know that when you don't have ciliated mucosa, like in Cartaginer syndrome or cilia dysmotility syndromes and cystic fibrosis, patients die because they cannot move the particulate matter out. And so understanding that, when the stem cell revolution hit in 2005, it was unbelievable. We had a laboratory for 25 years looking at tracheal transplantation. And when this hit in 2005, we literally closed our laboratory. We had several R01s, and we said, we don't need to do this because they're going to make little brains and intestines and a bone and, of course, trachea. And it was like overnight. This was on the cover of Time magazine, and they were talking about what the limits of this were. Now, organogenesis, making organs, and stem cell therapy are a little bit different. And what you see on the right here are using scaffolds, much like you've talked about, ear, trachea, and impregnating them with the stem cells to grow the type of cells that you want based on the properties that you introduce. And so literally with Yamakata's introduction in 2006 and the Nobel Prize for determining the ability to take skin and reprogram it into stem cells, there was no longer this ethical issue of using fetal tissue. And all of a sudden, this became the thing to do. And overnight, they were making ears in the backs of nude mice and valves and hearts, and of course, the simple tube, the trachea. And so it looked like it was going to change everything. And as you have all hit on, in this particular way that it's done, we take the patient's skin biopsy, we reprogram the embryonic cells, create the stem cells, grow them up, and then put them into some form of scaffold that you would put into the patient. And it's a process. The scaffolding can either be synthetic or biological. And they're the two different types. So in general, the concepts are that you may take your scaffold, you can either in vivo place the cells into the scaffold and place it right into the patient, or you can use tissue modifying factors, place it into the patient. Or what you can do is you can take in vitro the cells on a scaffold, place it into a bioreactor where you allow those cells to grow up and differentiate, and then transplant it in. So this is a staged approach. The first approach is a single modality for kind of tissue engineering airway. The synthetic types of scaffolding, usually they're propylene meshes or POS or PCU, and they look something like this. They're non-biological. And there is good data in the experimental world looking at this just in 2020, using a scaffold, the ability to not only create an outer matrix of cartilage, but an inner matrix of epithelial cells. The little bit of the problem here is that, you can only do small patches. You can't do this for long segments. So it's kind of our short mandible, long mandible problem here. You can take this avascular scaffold, put the cells on, but you really can't use it for extensive defects. And in fact, when it was attempted in these extensive defects in 3 patients, every single one of them died. And they died because you're looking at the endoscopic visualization. You have a long segment that may have started out with viable cells, but because it's not vascularized, it's not functional. These things clot, they clog, and the patient inevitably suffocates. And so this is a very, very common kind of resounding experience that they've gone through. So again, synthetic scaffolds like polymers, the problem is that they don't biologically integrate. They don't have vascularized components to support the epithelium. And so that has been a major problem with this form of kind of tissue engineering. Now, biological, they use decellularized human trachea, and we played with this for many years in fresh aortic grafts. And these would seem to be a little bit better. And again, if you go to the lab work, Chen-Yang Li and his group demonstrated you can take in a rabbit small patches and you can grow up epithelium. You can take it from the nose, you can plate it, you can grow it, you can put a cell sheet on, and sure enough, you can plate that little piece in there and get a patch graft. The problem is that, again, you can't do this for the extensive defects, the ones that really are problematic. And in fact, many of you know about Paolo Macchiarini and Martin Birschall. The two of them had created this approach of taking stem cells from the hip, injecting them into a scaffold, trying to grow up these cells and planting them into the patients. And as we all know, this led to not only retraction of all of their science because they never were really able to demonstrate epithelial cells and cilia motility, but also all of the patients that went through this transplant died. And we were actually up in Boston at the ATC meeting in 2005 when he presented this work, and we stood up and said, we see no evidence of ciliated cells, and kind of picked up on this, but there was a lot of blind eye to this. And again, Martin Burchell, attempted a very similar thing in a 15-year-old girl just in the last several years, taking cells, putting them into a bioreactor, trying to grow them up. And again, this 15-year-old girl died, trying to, learn the lesson that if you don't have a vascularized bed, you won't support epithelium, and these long segment tracheal defects just simply aren't functional, and the patients will clot off and they'll die. So again, the biologic scaffolds are interesting, but decellularized, whether it's aorta or human trachea, problematically don't grow ciliated epithelium. They're non-vascularized, and they tend to collapse over the course of time. So it leaves us with not much, and a brilliant approach kind of was taken by Pierre Dallaire and his group in Belgium. And what Pierre did was he said, well, fine, we're going to take trachea that's donor trachea. We're going to place it into the arm and let it revascularize. And then we're going to take buccal mucosa and we're going to line the trachea with buccal mucosa. And then we're going to take that trachea after 6-7 months, and we're going to take that graft and we're going to take it out in like a radial forearm and implant that. And Pierre's work proved effective. It takes about 8 months to do. It's buccal mucosa, so it's not ciliated mucosa. It works for larger patch grafts, and it's brilliant work. And Pierre has really pushed that forward. As vascularized composite allografts really became accepted in 2014 through the OPTN, and hand and face were being done, and we saw Maccarini's work fall apart, we reopened our lab and started a human tracheal transplant model. And we had used research over the past 25 years to re-explore this as an option. And to make a long story very short, we identified a way to revascularize the trachea, leaving in a piece of esophagus where the inferior thyroid artery actually passes. And we could take from the cricoid all the way down to the carina, 11-centimeter segments. And in fact, we performed our first human tracheal transplant in 2001. But in our mouse work, what we found is that the area that rejects in a trachea, you can see in this nice green pattern, is actually the epithelium. The T cells attack the epithelium. And so when you transplant in this pink segment to trachea, what initially happens is the donor cells start to die off, and the basal cells and the epithelial cells. But then interestingly, the basal cells and the ciliated cells of the recipient grow into the trachea, and you form an immune chimera or chimerization. And so now this graft becomes a little you and a little me. And we found this in the scientific work that we had done. And sure enough, when we transplanted our patient in early 2001, actually January 13th, and the patient did extraordinarily well, sure enough, this patient's epithelium had repopulated with the recipient. And this is important because hands and face transplants, you may or may not know, 85% of them reject. 50% reject acutely and 85% reject chronically. In our tracheal transplant, the patient did not reject at all. There was no even minor rejection. And we believe that the epithelium and the migration and the chimera that formed was really critically important. And so this plays into where do we go in the future? Now, the advantages of tracheal transplant is that it can work. We can do it. But the disadvantages is technically challenging, and it requires lifelong immunosuppression. But what we gained from this was an understanding that maybe we could marry the concepts of transplant and tissue engineering together and come up with something that was, maybe a little different. So there are two camps. There's the transplant organs, and then there's the stem cell people, our laboratory is really focused on marrying these two concepts. And so what we do with Yawen Chen, who leads the group, is we take trachea, we remove these cells that are the source of immune rejection, and we place human basal cells, stem cells. And we can get those stem cells to grow, and sure enough, they will grow inside any trachea that we place them into. And so using that technique, we can take these cells, place them into the transplanted scaffold because it's vascularized, and get them to grow beautiful ciliated cells that are functional. And so in the concept of how we see this, now using stem cells to inject into the trachea, or even through a inhalational approach, we now know that we can take human stem cells and repair injuries in the trachea from burns or intubation. So this is almost like doing a transplanted skin graft. But in the airway, these stem cells, when you inhale them, interestingly, they stick to the injured areas and they will grow. And so the place that we're looking to go in the future, and where we're really focused now in some new human trials that we're actually writing up, is taking the scaffold of a vascularized trachea, implanting the stem cells into it, growing it in, and then transplanting it into the long segment defects. The other approach would be to damage trachea, either from burns or acute injuries. We would take the stem cells and we would inhale them into the airway, where we've demonstrated that they will stick, heal, and grow. And so, the concept of regenerative medicine and scaffolding has been around for a long, long time, but the data on it just hasn't worked out. And our approach here is to say, why hasn't it worked out? Well, the scaffolds they use are not vascularized. So what we've done is said, transplant's probably not the way of the world, but what might be an interesting approach is to use that vascularized scaffold to marry the stem cells. So it's a big group. We coordinate our research with Columbia, USC, Stanford, UCSF, and others. Yawen Chen is the brains and really the one who runs this whole program. And I really owe all of the accolades to this group. I merely present the work and then try to explain it in English. But thank you very much, and happy to take any questions.
Wow, what a great talk, Eric. I did want to say I did get notice from the editor or the people that run the seminar that tell me that there's over 6,000 people watching, which for someone that's used to talking on reconstruction to 8 people in the crowd, that's astounding. So
Yeah.
So Eric, when you're doing your combined, or when they're doing this theoretically, is there any immunosuppression involved?
So when you take a scaffold, because it's a vascularized scaffold, yeah, you immunosuppress while you let those cells grow. And then you can, because the epithelium that would normally be the site of rejection has been replaced with autologous. So if we, God forbid, we're going to transplant your trachea, we would take a donor trachea without epithelium. We'd put your own stem cells into the trachea, grow them up, lay them in, let them grow, and then transplant that in. To give you immunosuppression for the scaffold is much lower and much more tolerable than the full triple immunosuppression we would use with the formal tracheal transplants that we've done, which is the same for a kidney transplant. And so the idea here is, one of the big targets is kids that are children that are born, that you don't want to keep them on that heavy dose of immunosuppression for life. So this would allow us to put them on a much lower dose, because the target of rejection is not the trachea, it's the epithelium. So you can use a much lower dose of immunosuppression.
And you're implanting that into the forearm?
No, what we do is we take the trachea now, that was one of probably the single biggest contribution I think that our group had made. We now can take a trachea from a donor with the inferior thyroid artery and the muscle and transplant it directly in. It doesn't have to go into a forearm. It's a single staged, transplant.
Fascinating. Anybody else in the crowd?
Incredible work, Dr. Gendon. It's I mean, it's a long ways from going from reality. It's come forward, right? You're part of a trying to transform that trachea, which is, I mean, with the cartilage was so hard to regenerate. I mean, even with an envelope, we have from a standpoint, what we in our shot presented is we have a natural soft tissue envelope, which is our capsule for vascularity, and we have neoangiogenesis forming into the bone. I mean, from a trachea aspect, it's really a vascular surface, hard to regenerate that. But kudos to your team for doing this.
Well, you know, the way we look at this research is that, look, tracheal transplants and this type of work is not something that's going to change the world necessarily. The idea behind it is looking at the science and saying, you know, what are we learning and how can we apply it to all the work that we do, right? So that's really where the people that have actually benefited the most from this are lung transplant people. They take our research and they say, you know, our lung transplants reject at extremely high levels. Maybe we need to spray in stem cell epithelial cells to reline the lung to prevent rejection. You know, we look at this in terms of mandibular, tongue, cheek, face, all the things that we could be doing and start to think about in tissue engineering, even in the mandible. You know, is there a role to take stem cells in a cadaveric mandible? Back in 2002, we took a cadaveric mandible and we placed in the donors, the recipient's stem cells through the marrow and tried to get that to take, and it took. It didn't take, you know, for 5 years. It eventually resorbed. But the point of this research is not because we're all going to go do tracheal transplants or stem cell trachea, but it's really to say, you know, what can we learn from each other? And how might that push us forward in the areas that you're interested in, for example?
Absolutely. And the one thing it was brought up when you mentioned that was some of the immunocompetent bone the companies have been using now. I've noticed it works very well in small defects, but in large segmental defects, I've used it. We have aseptic infections causing an immunogenic response. I think from a bone formation, a little bit of inflammation causes calcification and works great, a little bit of sample, but I've used it multiple times, 3 times actually, I can count. All of them cause immunogenic response from whatever competent proteins within that bone cells.
Yeah, yeah, it's an interesting area. The immunobiology of it is something we have to tackle. I don't think that, you know, stem cells and tissue regeneration, organal, you know, we build a lot of organs in our lab. We build little mini lungs and we study, you know, for example, how does COVID affect those lungs? It's human lungs. So we'll take your tissue and build a mini lung out of your tissue. So we know how your body would respond, say, to the virus. But using all these different techniques that Yawen Chen is really the pioneer on, she's absolutely amazing. We start to ask ourselves, you know, how could we affect mandible, you know, reconstruction, ear transplantation, you know, a bunch of different areas. So it's always interesting to talk with people, hear what they're doing, and essentially steal ideas.
Absolutely.
It's good
Perfect.
science.